This paper will discuss two related sets of experiments designed for establishing morphological damage datasets of characteristics found on apple seeds as the result of various processes performed on the fruits. By applying experimental archaeology to archaeobotany, it is the purpose of this research to assist in the interpretation of archaeobotanical apple finds, and thus to aid in understanding the role of apples as an economic and ornamental crop, and in its processing and consumption as food and drink. The results of the experiments will be applied to interpreting charred apple finds of the Middle Neolithic site of Oberderdingen in Germany. This presentation will focus on the experimental setting, its design, execution, and outline the methodologies for analysis. In 2019, a series of experiments was run at the York Experimental Archaeological Research Centre, or YEAR Centre, to establish the predepositional primary damage characteristics on apple seeds relating to specific actions performed on the apples, as part of a larger research project focused on cider production in archaeology. At the time of the experiment's design, the focus was on mineralized apple seed deposits found in the archaeology, such as the Roman occupation site of Doncaster in England. The Doncaster pit contained only mineralized seeds. The second largest quantity of seeds present in the pit were 23 plum stones, while the largest quantity represented were 1,400 crabapple seeds. This series of experiments was also heavily focused on cider production in the Anglo-Saxon period, prior to the Norman conquest of 1066, and how to establish the archaeological signatures usable to trace such production while combating issues relating to the production methodologies largely resulting in the use of invisible majority material culture and material culture possessing multi-lifeway object biographies. This series of experiments was broken into three main groups. The purpose of the first set of experiments was to produce used-word traces on apple seeds with possible Anglo-Saxon milling methods. Three such milling methods were selected: mashing with a wooden pestle in a wooden box, mashing with a stone pestle in a wooden box, milling with an iron nail inclusion drum grinder with a built-in hopper. The experiment type was predepositional morphology simulation, with some elements of process and function, as outlined by Outram in his 2008 Introduction to Experimental Archaeology. These were run as actualistic experiments. The second series of experiments focused on simulating alternative causes for the change in seed morphology to contrast with possible cider production morphology. The 5 methods used included simulation of kitchen waste via 3 different cutting methods, the simulation of human mastication and partial digestion, the simulation of unchewed livestock consumption of whole seeds and partial digestion, livestock partial digestion simulation of feed from the cider production byproduct, mash, and whole untouched seeds as a control. It is important to note that the number of variables for livestock mastication will need to be studied separately. Thus, the livestock mastication methods outlined here assume no secondary or tertiary mastication, and only partial digestion of the consumption of the mash. The partial digestion simulations were conducted by submerging the seeds in 50 milliliters of 0.1 molar hydrochloric acid for 5 hours, rinsed with distilled water and air-dried. The whole untouched seeds used as a control were carefully harvested from the apple cores to preserve the untouched seed morphology for the control. This was accomplished by cutting down the apples to their cores and submerging the core box in water. Gentle pressure was then applied to the core box with the thumbs, and the seeds removed with the fleshy parts of the finger pads. The seeds were then set to air dry. The experiment type for this batch of experiments was run as a predepositional morphology simulation and as a controlled experiment. The third stage of this experiment was to analyze the morphology of the seeds and identify if traces of cider production and other processes could be made, and thus to establish cider production archaeological signatures. This was achieved through micro-wear analysis and by establishing damage characteristics and their definitions. Primary and secondary damage characteristics were identified and the data quantified. This project resulted in identifying and establishing definitions for the damage characteristics for apple seeds. For example, a split is the separation along the fibers of the surface of a seed that breaks through the seed coat to the cotyledon, usually containing fibers either connecting to both sides of the split or broken at the inner edge of the split. A split can occur between two closed points or be drawn to the edge of the shell, creating an opening between one closed point and one open point. Due to some unforeseen external complications that resulted in the reduction of the statistical sample size, the data yielded from the partial digestion simulation segment of this experiment was rendered incomplete. These pie charts represent the ratio of prevalence of damage characteristics per action sample out of 100 as mutually exclusive representations. The damaged tip characteristic is highlighted in yellow. The large quantities of damaged tips for each of the actions may be contributed to flotation in separation of the seeds from the pulp rather than due to the production action itself. The post-action processing was different for kitchen waste than it was for the other actions, and this may contribute to the drastic reduction in tip damage for the sample. A controlled study on procedures may be required to make a final determination regarding the damaged tip characteristic. This chart illustrates seed damage representation per action type. The y-axis indicates the percentage of the represented sample where the damage characteristic was observed. It is important to note that one seed may hold more than one damage characteristic. 90% of all the seeds in the chewed seed sample presented fractures, while 63% of the chewed seed sample were observed to have splits present. Therefore, some of the chewed seed sample would have both splits and fracture damage characteristics on the same seed. The pie charts here show the chance of a specific action having taken place based on the damage characteristics found. For example, based on the data collected, if a seed with a pressure splinter was found, there is a 75% chance that it represents chewing, and a 25% chance that it represents kitchen waste. In 2000 to 2001, a partial excavation was carried out on a pitted site in the town of Oberderdingen in the southern German state of Baden-Württemberg. The pit with the charred apple remains is Middle Neolithic. The apple remains themselves were found in a cooking vessel. This vessel is the only one on the site to be identified as belonging to the style stage of Mk4, or the Michelsberg Culture 4. At the time of dating in 2019, this was the only Mk4 pottery to have been directly dated in Baden-Württemberg. The cooking vessel was found upside down, with the bottom broken. Sent for restoration, it was discovered that the vessel contained charred apple remains. The remains consist of fragments of apple skin, flesh, cartilaginous endocarp, and seeds. The sediment in the vessel was excavated and floated in 5 planum stages. Apple remains were found to be present in OBE 4, 3, 2, and 1. This experiment was designed to help identify the processes the apples underwent in the Mk4 pot. There are 3 primary purposes for the charring experiments. The first is to produce wear morphology on apples and seeds with possible Neolithic milling methods. The second is to produce intentional and accidental charring morphologies on the apples and seeds. The third is to produce accidental damage characteristics from cooking or storage accidents on the apples and seeds. These are designed as combined experiment types of process and function, that is process-focused, and predepositional morphology simulation, being run as controlled. The experiment itself can be broken down into 5 primary areas of focus with overlapping processes. The first series requires mashing the apples and charring with the juices. The second series requires mashing the apples, hand pressing the pulp with a cheesecloth to remove the juices, and the remaining pulp being charred. Series 3 chars whole apples where actions will be performed after the charring process. Series 4 is the focus test, and the 5th series performed is a fermented char. Of the processes requiring mashing of the apples, each will include batches of briefly mashed material and thoroughly mashed material to account for the processing variables. All experimental series will have separate batches for the charring variables. Low temperature charring to be run at 230 degrees Celsius and high temperature charring at 290 degrees Celsius. As this experiment is being conducted as controlled, the charring process is using a laboratory oven. The first 3 series have batches further divided by exposure. Each will have batches charred with full exposure to normal oxygen levels. Covered batches will use sand to limit oxygen and to illuminate charring post-collapse of a building or a vessel. Series 1, 2, 4, and 5 will have batches run separately with a wooden pestle and a stone pestle, used for the milling process to account for the materiality of related material culture for the production process. When it comes to botanical remains sourced through sediment samples, flotation or water sieving is commonplace. Series 1, 2, 3, and 5 will be assessed after flotation to account for the impact of archaeological processing of the samples. As not all samples are run through flotation, the experimental material will first be assessed prior to the process. The purpose behind charring whole apples is to account for post-charring actions. These actions include the collapse of a building or the ever-present soil shift as it impacts the archaeological record. To simulate the pressure placed on the charred apples due to shifting soil in the archaeological context, the apples are covered in sand. A board is then placed on top of the sand. Pressure is placed on the board with shifting weight to move the sand and displace the pressure onto the apples. For simulating the collapse of a building material, the same process will be conducted without the sand. The focus tests are conducted as a baseline for analytical control of the acute impact of the charring process on specific structures. Charring at both the low and high temperatures, the focus test looks at both the seeds and the charring of other prevalent anatomical features of the apple, such as the cartilaginous endocarp. Using the water box method from the previous cider experiments for harvesting unscathed seeds from the apple, the seeds are separated into 3 groups: whole untouched seeds for charring, whole seeds damaged prior to charring with wood and stone pestles, and whole seeds charred in an untouched state and then damaged. The fermentation char is being run as a minor eventuality trial. Batches of apples are separately milled briefly or thoroughly with stone and wood pestles and will be fired at low and high temperatures. The mash will remain with the juices while the juice ferments. Assessment requires a short fermentation period of 2 weeks and a longer fermentation period of 4 weeks. After fermentation, the juice will be hand-squeezed through cheesecloth to separate the pulp for charring. It is questionable if the ethanol will impact the seed coat. However, prolonged soaking and bloating of the seed tissues may react differently to the charring process. A special focus will be given to analyzing the cellular integrity of the seed coat's many layers and the cotyledon. At this stage in the project, analysis is in progress. Analysis is being approached through metrics, morphology, macro, micro, and SCM observation. The data will then be quantified and collated for processing. The weight and volume of the apples prior to charring and after charring has been recorded to assess charring shrinkage of the experimental material for each of the previously outlined conditions. Through the macro, micro, and SCM, observations will be collected relating to the wear morphology, charring morphology, and primary damage characteristics. Morphological changes to the fruit will be assessed by distortion and cellular integrity, damage or exposure of certain layers of the seed coat. Special interest will be paid to the cross-sections of broken edges and the variability of their condition due to the specific parameters of each batch produced: process, state of charring, exposure, level of contact with sugars, and the absence, presence, preservation, or distortion of the cotyledon. The research conducted at the Year Center was a positive start to this line of inquiry into cider production and how we can trace it and identify its archaeological signatures. It would be beneficial for further research to be conducted with larger statistical samples. Additional experiments conducting eventuality trials and mineralization would also be a valuable continuation of this research. Applying this research to the apple finds of the Oberderdingen site required expanding the research to include charring morphologies and other apple byproducts. An additional set of experiments were conducted to account for the influence of charring on the damage characteristics of the processed apples and especially their seeds. The charring experiments, although designed with the specific conditions of the artifacts from Oberderdingen in mind, will be of value for the interpretation of charred apple flesh and seed finds of other sites from other periods. Analysis is currently in progress, and the results will be released in the near future. I would like to say a big thank you to EXARC, the EAC13 volunteers, and Nikolaus Kopernikus University for organizing, hosting, and making this conference possible. And a thank you to Dr. Lena Maranova, Dr. Ute Sedel, Dr. Amy Little, Dr. Stephanie Wynne-Jones, and Dr. Andy Needham for your guidance and support in various stages of this research.